We're all in record now. So the DAMPs and the PAMPs trigger inflammation in a very non-specific way. But it's always ready to go. So you could say always on. And that's in contrast to the specific immune system, also called the adaptive immune system, where it takes several days for the adaptive immune system to be ready to fight. So it takes time to activate. But once it's activated, it's very specific and very targeted to the microbe that's there. So we're going to learn how that specificity arises and why it takes a little bit of time to get there. You've probably seen this outline before, showing you the cells of the immune system. It all starts with hematopoietic stem cells. Where do these stem cells live? Bone marrow, right. So every cell of the immune system is born in the bone marrow. There are circulating stem cells, and that's kind of a new field, and they're starting to be able to isolate those stem cells for stem cell transplant, rather than having to go into the bone marrow and harvest bone marrow stem cells. But the vast majority of your hematopoietic stem cells are in the bone marrow. And those stem cells, after they go through their first round of cell differentiation, could either go down a myeloid lineage or a lymphoid lineage. So the myeloid cells are going to become your platelets. So it can become megakaryocytes, which break apart into platelets, also known as thrombocytes. It'll become your red blood cells. Red blood cells are not part of the immune system, but they are derived from a common precursor. Mast cells, basophils, eosinophils, and neutrophils, we already talked about those types of cells. They can be present in both acute and chronic inflammation, but they are part of the innate immune system because they're not specific. So basophils, eosinophils, and mast cells tend to be activated by extracellular large pathogens, things like worms or big parasites. As we know, the neutrophils are your phagocytic expert, and they're going to come in with any type of inflammation, any type of acute inflammation. So any type of tissue damage, any type of pathogen will bring in neutrophils. And then monocytes are circulating in the blood. So they're born in the bone marrow, they go circulating in the blood, and then they pick a spot, and they go into the tissue and become a macrophage. So your macrophages are living in all of your barrier tissues all the time, ready in case somebody tries to invade. And they're also doing cleanup duty. So they're always kind of cleaning up. Cells are constantly turning over and needing to be removed. So that's what macrophages are doing. And then one other cell type that we haven't talked about for the innate immune system too much is this natural killer cell. It actually is derived from the lymphoid side, but it's not specific. So just because it is a type of lymphocyte, it is not specific, and it's part of the innate immune system. Its main job is to detect foreign or infected cells, cells that are infected with some other kind of pathogen, and kill them. That's why they're called natural killer cells. But in the last decade or so, there's been a lot of research about the role of natural killer cells in cancer. And a lot of the newer cancer drugs actually stimulate natural killer cells to try to get your body's own immune system to eliminate that cancer. So they're a really important cell type. But we're going to really focus over here today, T cells and B cells. So these are both lymphocytes. And they do the humoral immunity, are the B cells. And then the T cells are the ones that do cellular immunity. in a very specific way. So before we get more into that, I just want to review the different tissues of the body, because it's important to know how the immune system functions as a whole. So we've already talked about bone marrow. So bone marrow is where the stem cells and all immune cells are born. Bone marrow is also where your B cells are going to mature. And we'll talk about what that maturation is, but that's where B cells start to mature before they go out into the bloodstream. So your T cells are also born in the bone marrow, but then they go to the thymus to mature. So the thymus is this little organ right here where all of your T cells go through this process of self-tolerance. So you have to eliminate any T cell that detects a self-protein, and that happens in the thymus. So those are considered the primary ones because that's where your immune cells are maturing and differentiating. The secondary lymphoid organs are where your immune cells hang out and congregate. It filters the lymph and filters antigens. So these are things like the lymph nodes. And all of you have felt your lymph nodes before in your neck, right? So if you have a cold, your lymph nodes get swollen. And that's a sign that there's some activity going on in your immune system. So the lymph nodes are little organs where lymph comes in, blood also comes in. So lymph, you remember lymph fluid? So quick primer from anatomy and physiology, your extracellular fluid that's bathing all of your tissues drains into the lymphatic system, which is a parallel system of vessels, and it drains that fluid through the lymph nodes, and it eventually drains back into the venous system and recirculates with the blood. So it's basically a flushing system for the body. So lymph and blood both come into these lymph nodes. And this is a place where B cells and T cells are going to hang out. And it's kind of like a meet or greet. And sometimes, let's say that you had an infection in your skin, some of those macrophages and dendritic cells that were activated by that infection are going to travel through the lymph into a lymph node where it's going to meet some of these B cells and T cells and say, hey, I found something that shouldn't be here. Are you interested? And the B cells say, yeah, I recognize that. And that's going to activate them. So the lymph nodes are kind of like a meeting place for cells of the immune system, and they're a filtering place. So from the lymph fluid, you might have macrophages or dendritic cells. But you could also have microbes or particles that are antigenic coming through. So then all of those things are circulating through this place where there's a lot of B cells and T cells. If a B cell or a T cell gets activated, it's going to undergo clonal expansion. That also will happen in lymphoid tissue, such as a lymph node. These mucosal-associated lymphoid tissue down here, MALT, sometimes you'll hear it called GALT, gut-associated lymphoid tissue, and specifically, you'll see them on histology referred to as Peyer's patches. So these are like lymph nodes, except for instead of being separate from an organ, they're embedded in the wall of the mucosal system. But it's the same thing. It's where lymph fluid drains through and B cells and T cells hang out and meet up with other cells of the immune system and can become activated and proliferate. And then the spleen is actually just filtering blood. So there's a lot of lymphocytes in the spleen, but it's looking for any antigens or microbes that are in the blood specifically, rather than draining from your tissues. That's why it's really important for people who have their spleen removed to get vaccinated against a lot of different pathogens, because they're now much more susceptible to bloodborne pathogens. And the spleen is also, it removes debris and dying or dead red blood cells, so it's another filter organ. Any questions? So you've already seen this timeline, and we've already talked about the innate immunity. Again, your barriers keeping microbes out. If they do get in, you'll start to phagocytose them. Do you remember complement? So I mentioned complement. It's made in the liver. It's a system of proteins. There's more than 20 proteins in the complement system that are constantly circulating in an inactive state, and they can be activated by inflammation. But you'll see it'll also come up that they play a big role with antibodies and humoral immunity. So complement is part of the innate immune system, but it can also interact with the specific immune system. But again, this happens really quickly. This is showing you a timeline. Some of these microbes are not going to be phagocytosed right away. They might be carried through the lymph, and they might be activating B cells. Or one of these phagocytes will carry and present to one of these B cells or T cells. So the microbes, as soon as they get in, they're also being presented to your lymphocytes to start the process. Most of the time, your innate immune system clears it before you've even gotten to the point where you're making the effector cells. Why would you still want to make them? Any ideas? In case you see it again. So your adaptive immune system, your lymphocytes, have what we call immunologic memory. They can remember. So you can fight this, but you want to be able to fight it even faster the next time it comes, because what if the innate immune system doesn't do as good a job next time? So the adaptive immune system plays a really important role, even if it doesn't do the first line or second line of defense, in keeping your body safe over time. So here are the key features that you should know about the specific immune system. First of all, it's slow. So it'll get stimulated right away, just like the innate immune system, but the innate immune system is ready to go while the adaptive immune system needs time. So it takes 3 to 5 days from the initial stimulation till you get effector function. And the reason it takes that long is because it's highly specific. So each lymphocyte recognizes only one antigen. Now, a bacteria, what's an antigen? Can somebody define an antigen for me? A surface protein? That is, the majority of antigens are surface proteins. But an antigen is really anything that can trigger the immune system, to kind of make it full circle and confusing. So an antigen is usually a protein, or actually a peptide, I should say, because it's smaller than a protein, or a carbohydrate, or some combination, some glycoprotein, like a protein that has a little carbohydrate group on it, or a lipid that has a little carbohydrate group on it. So it's a specific size and shape that is recognized by the immune system. That's an antigen. Most of the time, they're proteins on the surface of something, but it could also be carbohydrates. So if Streptococcus gets into your body, it probably has on its surface 100 different antigens. And circulating in your body all the time are B cells and T cells. Each one recognizes only one antigen. So of the tens of thousands of cells, lymphocytes circulating in your body, maybe 50, maybe 100 will specifically recognize Streptococcus and not any other type of microbe. So very specific to those antigens. Memory. Once your B cells and T cells have been triggered by a specific antigen, they continue to remember that and divide. And so if you were infected by chickenpox as a kid, your B cells that recognized chickenpox continue to circulate through your body and continue to make antibodies to chickenpox all the time. Even as you get to be 50, 60 years old, they're still doing it. They remember, and they never want you to get chickenpox again. So that's what it is. And that's the basis for vaccination. So a lot of you probably got the vaccine rather than got chickenpox. Is that true? And so when you got the vaccine, they gave you an attenuated form of varicella virus, one that can't cause disease, but it still has the same antigens as chickenpox virus, as the active form, and so it triggers the same immunologic memory, so that if you ever are exposed to that virus later in life, you won't get the disease. Your antibodies are already there and ready. Yes?
What happens if you get chickenpox twice?
If you get chickenpox twice, it just means your immunologic memory was not very good. Is it bad for the future? So the problem with chickenpox is that that virus never goes away completely. It hides in your nerve cells, and it travels up into your spinal cord, into your dorsal root ganglia, and it hangs out there. And every time it tries to start to divide and get back into the blood, those antibodies say, you're staying put, and it won't let the virus divide. But if your immunity goes down for some reason, what do you get? Shingles, yes. So as you get older, your immunity starts to wane. You have fewer immune cells, and they don't work quite as well. So you're more likely for that virus to continue to come out. So the biggest risk of getting chickenpox twice is that now more of your nerve cells have that virus in them. And so you're maybe at increased risk of getting shingles. And maybe you want to have yourself tested for an immune deficiency. No, I'm just kidding. Some people, for whatever reason, some people may not develop a full immune response the first time. That's why if you do get vaccinated, they recommend boosters to help keep that memory sharp. And then this idea, the last idea, is self-tolerance. So you don't want to specifically recognize antigens that are part of your own body. So you have to eliminate any lymphocytes that recognize self-antigens. And when that process doesn't work well, you get autoimmune disease.
Do they just spray them in PCM around the cells and then they turn it?
Yeah, so right with this. Okay, this is what I wanted. So let's go back here. So we had our B cells, we had our cytotoxic T cells. We also have these helper T cells. So what do? They actually activate both the cytotoxic T cells and the B cells to do their effector function through the release of cytokines. So you don't have to memorize all of the cytokines that T cells use, but you remember that cytokines are chemical messengers for the immune system. And so T helper cells, when they're specifically activated, are going to release cytokines that interact with the other cells of the immune system. And if you remember back when we were talking about chronic inflammation, that macrophages, after the acute inflammatory process, if there's an ongoing stimulus, they're going to kind of change their cytokine profile to trigger some of these T helper cells. And those T helper cells are also going to secrete different cytokines to change the role of macrophages and to recruit other cells like eosinophils or basophils. So the cytokines that are secreted by the T helper cells help to coordinate a bigger immune response, and the T helper cells are critical for activating both B cells and the cytotoxic T cells. So they're aptly named because they really do help the immune system in many ways. And this is just a little pictograph showing you that process. So I like pictures, it's good, I want you to go back and review it. Are there any questions about these two arms? Yeah.
On that last one, did you say that
So the cytotoxic T cell, and let's see if it's on this picture right here, will get up really close to an infected cell. In fact, it knows that cell is infected because a receptor on the T cell binds to a signal on the infected cell. And so it has to be right up next to it. Those two cells basically are bound together. And then it releases these chemicals directly onto that cell. And basically what it's doing is it's poking holes in that cell and activating those caspases. Yes?
Do the helper T cells have any chemical mediators?
Yes. Okay. So how am I doing on time? Am I getting carried away?
There's 25 minutes.
25 minutes left. Do you guys need a break? Oh, just a couple minutes? 2 minutes break? Okay. So as we get further into the immune system, go back to that and really keep in mind, what is humoral immunity all about? Why is it doing this? What is cellular immunity all about? Because there's a lot of details and it's going to get very confusing, but trying to keep those big pictures in mind will really be helpful. So let's move into each of the different types of cells. We'll talk about B cells first. And what they do. So B cells, first of all, they are born in the bone marrow. We know that. They mature in the bone marrow. So when a B cell is born, it has a receptor on its surface. And that receptor is called a B cell receptor. And the B cell receptor is designed to recognize an antigen. The B cell receptor is actually an antibody anchored into the cell membrane. And it recognizes a specific antigen. In the bone marrow, it's tested. Does that B cell receptor recognize any antigen that might be found in the body? And if it does, that B cell is killed by apoptosis. So that's what we mean by maturation. And that process for B cells happens in the bone marrow. So after they're born and they're expressing the B cell receptor, they're tested for self-reactivity. If they're self-reactive, they are eliminated. If they pass the test, then they can go out. They're mature B cells, and they go out into the bloodstream. so they can circulate in the blood and the lymph fluid. And they hang out in the secondary lymphoid tissue. So remember, they've passed the first test, the test of self-reactivity. So now they're out in the body with their B cell receptor, waiting to see if any antigens match them. And since they passed the test, if an antigen matches their B cell receptor, that antigen is foreign, as long as the body's working correctly. And that foreign antigen needs to be eliminated. So that foreign antigen will stimulate or activate the B cell. So now, how is this process of activation designed? It's very complex, because what happens if you activate a B cell to a self-antigen? It's not good, right? You're going to attack your body, and that we don't want that to happen. So we've actually set up a 2-signal process for activation. So the first signal is that they have to be, that antibody that's on the cell surface, the B cell receptor, which I'm going to abbreviate BCR, B cell receptor, binds to an antigen. And we're hoping that antigen is foreign. It should be. But just to be sure, that B cell will not be turned on unless a T helper cell also stimulates it. And so that's called, the T helper cell provides the co-stimulatory signal, or signal 2. And that T helper cell had to be activated by an antigen as well. So it gets quite complex. But it's this 2-step process. And the point of that is to really keep the immune system tightly regulated. You want it to react, but you only want it to react if it's absolutely necessary, if you're absolutely sure that it's foreign and not self. And as we've already described, what happens once it's activated, it's going to start to produce antibodies. So this little diagram over here shows you this 2-signal process. So the first signal, here's that B cell receptor, looks just like an antibody. It is an antibody. So that B cell receptor binds to an antigen. If there's enough antigen, and you wouldn't just bind to one microbe, but if there's 1,000 microbes, these microbes are going to bind to these B cell receptors and cause them to cluster. That clustering is signal 1. It activates some other these transmembrane proteins and delivers signal 1 into the cell. It says, okay, we're about to turn on. But it's not going to finish that turning on unless there's a signal 2. So over here they're showing, there's actually another way that you could activate signal 2 that they're showing in this picture called complement. So this second signal will come from CD21, which is the co-stimulatory molecule being activated. So it can be activated either by a T helper cell in the area or by complement. And so that complement binding here, or more usually it's the T helper cell, and that's going to deliver the second signal. Both of those signals together turn on the cell and say, okay, let's start making more antibody. So this antibody that has been on the surface, called the B cell receptor, is a useful antibody. Let's make more of it and start to secrete it and put it into the blood. So it's going to, once they're activated, they start to make antibody. And it gets a little bit more complex than that. because you also go through a process of trying to tweak the antibody to see if it can be even better, if it recognizes that antigen even stronger. But we're not going to get too much into that in this class. But you may have heard about that and be wondering how it all fits in. So that's going to fit in there. So let's talk for a minute about antibodies. How many of you have learned about antibodies before? Okay, good. In micro or in other classes? Which classes? Okay. So you've learned about it in anatomy and physiology, infectious diseases, some other classes. So this is really just a review for some of you. Are there people who this is new information for? No? Okay. Don't be shy, because it's important to make sure that we all kind of are coming together here. So I'll be quick, but it is important to understand. So antibodies have, oops, let me go to my pointer here. A heavy chain, which is what you see here in orange. So they have 2 heavy chains and 2 light chains, which you see here in red. And those are bound together by disulfide bonds to act as one unit. At the end of part of the heavy chain and the light chain, where they're connected together, at that end is the antigen binding site. So this whole region is called the FAB portion, AB for antigen binding. This region down here, where it's only the heavy chain, there's none of that light chain, is called the FC region, C for constant. And what that means is that this region down here, the FC region, doesn't change or mutate, or it's not variable. The antigen binding region is going to be variable. And that's how you recognize such a diverse array of different antigens. And the way these heavy and light chains are formed is through a process of gene rearrangement. Have you guys heard of VDJ rearrangement? So there's a region on your chromosome that code for a series of genes called V, D, and J. And these genes can be spliced together in a lot of different combinations. So you have maybe 40 different V genes. And in every single B cell, you pick one of those V genes. So 1 out of 40. And then you have maybe 25 D genes. And so for every single B cell, they pick one of those 40 Vs, and they put it next to one of their 25 Ds. And then there's 6 Js, and they pick one of those Js. So you get a combination of VDJ that when you do the combinatorial calculation, there's 3.5 million combinations. Of the VDJ genes. So that's what codes for these heavy and light chains and gives you that antigen binding region. So every B cell has a unique antigen binding region. That's how every B cell can recognize only one antigen. And they're learning more and more about it, and it becomes even more complex. So that's where we're going to hold it for now. This constant region is another, it's a C gene at the end, and that determines your antibody class or type right here. So the FC can either be type M, so the M gene, type G, type A, type D, or type E. Those are your different types of antibodies. And what that means is that it's a different constant region of the antibody. And the reason you have different constant regions is so that your antibodies can do different things. So all cells are born starting with either IgM or IgD. So that B cell receptor that's bound to the surface of the cell, of the B cell, is either going to be type M or type D. Vast majority of them are type M. And so as soon as it's activated and starts to produce antibody, it's producing a lot of type M antibody. And type M antibodies are really good at activating complement, because they like to form these pentamers. So 5 IgMs will all get together, and then they'll activate complement. They can also bind to and opsonize the microbes themselves. But IgGs, those are the most common antibodies in your blood. So these are circulating. And they're really good at opsonization. Opsonization means the antibody binds to surface antigens on the bacteria and triggers that bacteria to be phagocytosed. And the reason they're so good at that is that they tend to circulate by themselves. IgAs tend to circulate in dimers, and their constant regions bind to this secretory protein that triggers them to be secreted into your mucous membranes. So they're part of the mucosal immune system. So IgAs are found in breast milk. They're found in the mucus lining your digestive system. So if you get norovirus, it's your IgAs are going to be secreted into the digestive system to try to opsonize that virus, to try to bind to that virus and keep it from getting in the body, or to kill it, to trigger your immune cells in the area to kill it. Does that make sense? Okay. IgG, not very common. It's one of the first antibodies. We're still learning about it. We don't really know what it does too well. IgE binds to mast cells and eosinophils. And so we're going to learn more about that when we get into hypersensitivity reactions, because IgE basically primes your mast cells to dump histamine and other granules when there's a trigger. So there's different types of antibodies. They do different things, but in general, these are the functions of antibodies. So neutralization of microbes. So the antibodies directly bind microbes. And you'll remember that microbes often use cell surface receptors as their virulence factors. So for attachment to your mucosal surface, you might have certain types of proteins on the outside of a bacteria. For entry and penetration into your cells, viruses use cell surface or their capsid proteins or envelope proteins, spike proteins. So when an antibody binds to those proteins on the surface, they're blocking attachment or they're blocking cell entry. So they can directly block the ability of that microbe to continue an infection. And that's called neutralization. Another thing they can do is once they bind to those cell surface proteins, other phagocytic cells in the area recognize the Fc portion of the antibody that's bound to that microbe and will phagocytose it. Now, we already talked about with the innate immune system that you can phagocytose bacteria without antibodies. But antibodies make the process more efficient, and as you learned in microbiology, if a microbe has a capsule, the macrophages may not detect it and may not be able to phagocytose it. So antibodies can actually help overcome that process. So antibodies increase the ability of phagocytes to recognize and phagocytose these microbes. So that's opsonization. We're not going to get into the antibody-dependent cytotoxicity too much here. It's very similar to what we talk about with the cytotoxic T cells. And then the last thing I want you to pay attention to, we have opsonization, is activation of complement. Now, complement, I've already talked to you about it because it's part of the innate immune system, but it also interacts with the adaptive immune system. So with the innate immune system, complement can recognize foreign cells like bacteria or protists, and it can form membrane attack complexes and directly kill those cells. But if an antibody is bound to those cells, specifically IgMs, which are really good at this, those antibodies can actually increase the ability of complement to recognize the bacteria. So similar to the opsonization increases phagocytosis, complement increases the ability of complement to be activated and kill those cells. So you can get lysis of microbes. Complement can also increase phagocytosis. And then I also told you that complement can act as a co-stimulatory signal for B cells. So there's a lot of roles for complement to interact here. So basically, you can activate complement through antibodies. And then part of that will also help with innate inflammation. So that's where we come back, and there's an interaction between the adaptive and the innate immune system. Questions? Okay, don't try to memorize this slide. This is simply to show you complement is complex, and it does a lot of different things. The one to look at here is this one right here. Antibodies bound to bacterial antigens can activate this complement complex, which is going to cause that bacterial cell to die. It'll kill the bacterial cell. So that's an important role for complement and antibodies in immunity. Are we out of time? 2 more minutes. I can do this. Oh, you know what? This is probably a good point.